My name is Aileen from Intan Permata School. I would like to present about my research about the effect of salt stress on the seed activation of Leucaena leucocephala. Sprouts contain many useful chemical compounds such as vitamin C and phytochemicals that may bring benefits for our health. In general, the germination process can be distinguished into three phases. Phase 1, where water imbibition occurs. Phase 2, involving metabolic reactivation. Phase 3, where cell elongation happens. As seeds imbibe water, they expand, increase in mass, and hydrate enzymes, which activate, and the seed increases its metabolic activities to produce energy for growth. The purpose of this research is to investigate the alpha-amylase activity in Leucaena leucocephala seed. This research is done by observing how salt stress influences the metabolic activity of the seed. The research questions are: does salt stress affect the time needed for the Leucaena leucocephala seeds to reach phase 2 of germination? Does salt stress affect amylase activities of the seeds' germination? The measure of the rise in metabolic activity was done by measuring the rate of amylase by observing and timing the change of color. Amylase was chosen to be observed due to its practicality to be detected. The first stage of the research was done by soaking the Leucaena leucocephala seeds in water as a control, and in NaCl water with different concentrations, 0, 50, 100, and 150 millimolars. Phase 2 of germination is determined by measuring the rate of the seed masses change every 24 hours. The seeds are said to be in phase 2 when there is no more mass growth for 48 hours. The change of the seed mass is observed until the mass is constant. After 6 days, all of the samples were crushed using a blender after being placed in the fridge for 24 hours to terminate further growth. The seeds were blended afterwards, then temporarily soaked in 2% NaCl solution. They were then filtered using filter cloth. The supernatant was taken while the residue was removed. The last is to conduct an enzyme test. The amylase activity is tested through its ability to fully catalyze the breakdown of 1% starch solution. In this research, Leucaena leucocephala seeds show mass increment from days 0 to 4, indicating that imbibition is taking place. After that, the seed mass will remain constant during phase 2 of seed germination. The treated seeds show higher mass increment and faster entry to phase 2. This rapid rise likely happens to the entry of NaCl to the seed cells, causing the rise of osmotic pressure in the cells, while the faster entry shows that the treated seeds have higher metabolic activities. Seed metabolism activation starts from the excretion of GA by the embryo, which then triggers the production of hydrolase enzymes. Amylase plays a role in changing starch to sugar. The activity from this enzyme can be tested by the iodine method. From the result of the research, it can be concluded that salt stress, which is an abiotic stress, does affect metabolic activity of the Leucaena leucocephala seed, which contributes in increasing the amylase activity rate. Salt stress increases the mass and rate of alpha-amylase activity of the seed in phase 2 of germination. Salt stress increases the seed activation of seed. So that concludes my presentation. Thank you.